All right, so let's start with the first lecture of the day, when I'm going to go through the current reference genome sequencing initiatives, and also will try to introduce you to all the best practices or things that you need to think about before you're actually starting, even considering sequencing a reference genome. So why are we sequencing reference genomes, and how did we come up with this beautiful idea that we need to sequence reference genomes? You know that the Sanger method was created and then published in late 70s. And then it took more than 20 years to produce the first reference of eukaryotic genome. And that was the Baker's yeast Saccharomyces cerevisiae. It took almost 20 years, 600 people from 100 different labs all over the world to produce the sequence data and also to assemble this genome. And here I introduced a couple of the QR codes that you can see on the screen. And one of them is the original paper when scientists discuss this genome and also the effort. And the second paper is more about the history of the yeast genome project. This is something that I enjoy reading from the perspective of science history, or scientific history. And this is something perhaps that you will find enjoyable read as well. So in the first paper, people sequenced the genome and they started to put it together. And in that time, we did not have this massive parallel library preparation methods or massive parallel sequencing, excuse me. And even we did not have something that is nowadays is called bioinformatics. So people had to do everything in small chunks and then put everything together by hand. And then the genome, when the genome was sequenced, it's needed to be also annotated and understood. So which A, Gs, Ts, and Cs code for what kind of genes and how are they arranged in the genome and what function they have. So, and scientists already there, they said that would be a global effort and that would take many, many people, many, many years to get anywhere closer to actually understanding what the yeast genome does. That was a long time ago in a galaxy far, far away. It was in 90s. And just in 30 years, look where we are at now. So currently in a single lab, you can sequence the yeast genome within a couple of days, and then to do assembly within a couple of days, and then to make an annotation. It might take longer time, but for something as simple as yeast, it's approximately 2 weeks work to do the entire workflow. So we sequenced yeasts, but why did we sequence yeasts? 1/3 of the yeast genes, they actually have a very high homology to the human genome. So yeast was studied to understand the basic cell functions. So lots of cell molecular biology that we are studying today and we have knowledge of, all this information is coming from the yeast genome. Then, couple of years later, the tiny worm or nematode C. elegans Cinerhabditis elegans has been sequenced, and C. elegans has been used for discovery of human disease genes. Okay? And then to study Mendelian traits and also link of phenotypes and the genotypes, people also sequenced Drosophila melanogaster, or the classical Mendel model organism. In 2000, there came the first human genome. It was quite fragmented, but it was Arabiopsis thaliana, and people wanted also to know how the plant genes are built, how they're regulated, how the plant cells are functioning differently from the animal cells and from the fungal cells. So they did that as well. But as you understand, it's very difficult to study human neurodegenerative disorders if you are studying yeast, or it's very difficult to look at blood malignancies when you are studying Drosophila. And it's also very difficult to find out how the human heart is functioning or what kind of genes are involved in these networks when you're looking at the C. elegans. So these genomes definitely were not enough. And then researchers, again, from all over the world, they decided that now it might be a time to create the reference genome for a human. Talks about that started already in the 90s, and then the Human Genome Project was launched later in 2000s. And people were thinking for a long time, is it worth it? What can we learn from human genome? Will it help us to do the science and the medicine? And they boldly decided to go where no one went before, and they decided, yes, we want to sequence the human genome. What did it give to us? To us researchers, the reason why we are here now and listening to this lecture, it gave us a genomic revolution. It created completely new technologies, what now we call massive parallel sequencing. It gave us the new field of genomics when we can look at gene regulation and also to evolutionary processes in a completely different level of resolution. It also created a completely new science called bioinformatics. So all those huge advances in medicine, huge advances in sciences, are actually thanks to those bold people who decided to sequence yeast genome, although it took 20 years. They decided to sequence a human genome, although the project itself actually is still going on, trying to decipher the telomere to telomere chromosomes. But human is only one species that inhabits this planet. And we're sharing that with trillions and trillions, and billions of different species. And we also want to make sure that the people who are cured due to molecular medicine, thanks to the advantages of the human genome information, that they have a planet for their children, and these human populations can sustain their life. And also we can have the same quality of life and live together with nature, and also get our food from nature, and so on and so forth. So we need to study nature, because currently we're facing something as a sixth mass extinction event, when we're losing species faster than we can discover them. So people started to sequence genomes and do all other types of biodiversity genomics. And the idea behind that is to create an open data repository. There are different kinds of sequencing initiatives. Some of the sequencing initiatives, they are concentrating on reference genomes only. For instance, the Earth Biogenome Project and European Reference Genome Atlas. There are initiatives that are sequencing the barcodes or certain portions of the DNA in the genome that could help to place organisms in the tree of life or identify them taxonomically. The examples of that, for instance, is the eyeball, International Barcode of Life. Other initiatives, they are looking at the eDNA, and they are trying to profile all the organisms that can be found in a certain ecological niche. There are several examples of these large types of consortia. Besides different applications, reference genomes or barcoding or eDNA, these projects are also in different kinds of scopes. They can be international. For instance, again, the ERGA, the European Reference Genome Atlas, spawning across different countries. They can be national. An example of that would be the Darwin Tree of Life, that is sequencing everything on the British Isles. They can be regional. For instance, a Philo-Alps project that is looking at the alpine populations, the alpine organisms in the center of Europe. Approaches can differ. They can be taxon-driven. Example of that is a project Psyche, when they sequence all the Lepidopteras or all the butterflies and moths, in Europe, at least for the moment. They can be application-driven. So, for instance, the USDA, United States Department of Agriculture, they have a program sequencing agricultural pests to understand what is going on. And also they can be ecosystem-driven. An example for that, for instance, is California Genome Project, and a part of it that is taking care of the coral reefs and looking at the coastal lines. And also that would be the Atlas Sea, the French initiative, which is sequencing all the French territorial waters and trying to understand what kind of organisms they are and how their genomes look like. All of these projects, they aim to create an open data repository. Why? We know that there is a biodiversity decline. We also know that the genomics and genomic data, they enable many disciplines in biology. But we know also that sequencing is still quite expensive. We know that individual groups And let's face it, the most of the sequencing is done in global north. Sequencing is not readily available in other countries. So these large consortia and projects, they want to sequence these genomes and put them in the open source so that other countries or smaller research groups, they don't have to do that. Within these projects also, we try to standardize the methods and standardize the metadata so that the outcomes of this project, this huge library of genomic life, it is fair, it is ethical, and it's also legal. But the main thing, after we started these huge projects, there is a very interesting thing that happened. Now genome became a tool. It is not a goal anymore. So when people were sequencing E. coli, you sequence single strain somewhere in the 90s, and you could publish that single strain in Nature with impact over 30, impact factor over 30. So now, without having a good scientific question, you cannot publish a genome. So in all these initiatives, we're producing tools that is up to scientists then to take these tools and apply them to understanding their biological systems, to answer their scientific questions. So that's a huge paradigm shift that we are observing now in this field of modern biodiversity genomics. The flagman of all these efforts, of course, is the Earth Biogenome Project, which is a network of networks. And they are trying to sequence all eukaryotic life, starting from higher taxonomic units, and going down to families, and then going down to genera, and then going down to species. So it's heuristic approach, hierarchical approach to sequencing life. And Earth Biogenome Project, it spans all over the world, and there are many smaller projects that are associated with this large one. So this is one of the projects I will be calling EBP, and it will be shown many, many different times, so addressed many different times within the entire course. The second abbreviation I would like to introduce you to, it's European Reference Genome Atlas, or ERGA. ERGA is a child of pandemics, when researchers from all over the Europe came together and decided, okay, so we have the Earth Biogenome Project in the world, let's do something European, because we knew that Darwin Tree of Life in the UK were extremely strong. We had guys in Catalonia who were sequencing their genomes, who had very strong German component, they were working on vertebrates. Here in Sweden, we were doing birds and other things. So many different institutions, many different sequencing centers, many different universities, they came together and created this network. And there is a QR code. You can pause and you can take a picture. It will take you to our homepage. And I have an honor serving as a current vice chair of this consortium. Then ERGA merged together with the International Barcode of Life, or IBOL consortium in Europe. And we decided to create one large European project which is going to enhance the capacity of both barcode and genome production in Europe. And that is something that is called Biodiversity Genomics Europe. And this project is sponsored by the European Union, the Horizon program. There is a QR code to look into it. The two streams, which is ERGA and Bioscan, or previously known as Bioscan, now it's known as the IBOL branch, International Barcode of Life branch, we're working together, bringing our forces to try to decipher European biodiversity together, both from the genome side and from the barcode side. And this is the representation of different participants in this project. And most of the lecturers who will be talking to you today and presenting, they are one way or another, they're connected with the BGE, or Biodiversity Genomics Europe, or they're connected with ERGA. So and within this project, we have officially established ERGA as a European node of EBP, or Earth Biogenome Project, and IBOL Europe, previously known as Bioscan, is now a European node of International Barcode of Life initiative. On the genome side, what are we doing in ERGA? we are using and also contributing to all the sets of recommendations at the Earth Biogenome Project level, but also we have a sets of committees and sets of working groups trying to create guidelines and standards for different parts of the flow of reference genome generation. It can be from sampling with involvement of museums, and this is something that you will hear from Paco's lecture very soon. That is sampling and taxonomy. Then there are also sequencing labs and also molecular biology labs, which are preparing recommendations for DNA extractions and also for DNA sequencing with involvement of the sequencing facilities. And finally, there are teams of bioinformaticians, and you'll have Thomas' lecture later today. He will give you more information about that. In terms of computational genomics and data handling, there are very strong teams of data stewards, and that is Niklas and Stefan, who are going to talk about that. And then finally, there are people, more scientists, looking at the genome analysis and trying to understand how it fits in the global perspective and evolutionary biology perspective. Within the BGE, the work that I'm going to present to you today is a joint effort by several different partners. So I'll be talking about work from so-called Work Package 5, which is the sampling and the vouchering. Next, there will be also talks about the sequencing initiative, and the sequencing work package is led by Sanger together with SciLifeLab. And then the assembly and annotation, that is SciLifeLab NBC team. The effort is also led by Sanger, but our bioinformaticians within SciLifeLab are very strong contributors to this common knowledge. That is the program. How is it going to look today? So we go through all the steps of the genome generation. Please pause, have a look, and read more. I will not stay here longer, but in this particular lecture, in my part, I want to focus on the recommended project workflow before sequencing. Because in the beginning, I promised you that I will tell you things that you should know before starting your sequencing project, or what we as sequencing providers and data analysts wished you knew before you started your reference genome project, okay? So this is something that crystallized out from the work that we are doing in ERGA and in BGE. First of all, when you decide to sequence something, we would urge you to check the latest sequencing recipe. Because, I will show you later, it's not that easy and it's a constantly moving target. After that, we would suggest that you get in touch with people who you intend to sequence your genome, just to check what kind of sample requirements and necessary paperwork is needed, because that can take a long time. And that actually is crucial that you do it even before going to the field, because the sequencing facilities could give you some advice to future-proof your sample for the reference genome sequencing. And that will be in one of the lectures later today. Then another thing is, it's nice if you start gathering information about how this work has been done in samples of the similar taxonomic groups. Check how the samples were collected, check how the samples were preserved, and that is especially important if the results of the reference genome sequencing is really good in that particular paper. So if you see that, oh my God, that's a really good genome, find out how they did it. Read materials and methods, and perhaps reach out to the authors. Usually, people really would like to help other people not to make the same mistakes as they did before. Extremely important point is to check all the permits, and that is something that is called Nagoya, access benefit sharing, and also shipment permits. After that, only after that, you go and collect the sample in the field. After you collect the sample in the field, you need to identify it correctly. Also, if possible, it's nice to include DNA barcode sequence. For instance, cytochrome oxidase, Orobisco, or something else, depending on the organism that you're working on. After that, you need to make sure that all the documentation for the sequencing facilities is in order. So when your sample will arrive to the sequencing facility, together with hundreds of other different samples from all other different people. Your samples, they will not get lost. So they are correctly marked, they are correctly placed, and so on and so forth. And then the sequencing facility actually knows that your sample will arrive on time. After that, you get in touch with the courier company, and maybe you need to be quite picky about the courier companies, and about that we'll speak a bit later during the workshop today. And then finally, only after all these steps, the step 9 is actually shipment to the sequencing center. And the layout, this layout is something that will be discussed in several different lectures. So sequencing recipe, I will introduce to you very shortly. The sequencing facilities and a little bit of the field collection I will speak about later today. Nagoya ABS shipment permits. Yuan, Sebastian, and Niklas will have a joint lecture about that, and then about the shipment permits and biosafety, there will be Henrik's lecture later. Collection in the field, collect taxonomical identification, also vouchering, is something that Paco is going to cover shortly. And then finally, we'll go through the steps at the sequencing facilities and also the courier companies and the shipment in my lecture in the afternoon. The reference genome sequencing recipes. It's a moving target. I started in the field of genomics of non-models as early as 2005. And at that time, we were doing everything on Sanger, and then it would be primary WOG and phosmid and cosmid libraries and BACS and YACS, you name it. But I started at SciLifeLab in 2012. And at that particular time, the golden recipe or the gold solution for reference genome generation, that would be Illumina, 100x coverage, and then on top of that, Illumina mate pairs. You don't know what Illumina mate pairs are? Go online, Google it, and try to read. It's archaic, but it's a really cool technique. Then, just a couple of years later, long reads came. So PacBio came first, and then came also Nanopore. But Nanopore was still very expensive and not used for something more than bacteria or fungi. So PacBio was very popular at that time just for reference genome generation. And we used to sequence 30x with long error-prone reads per allele. Then we would do Illumina scheme at low coverage to try to correct for those errors. And then there was the technology, 10x Chromium, where nowadays you do single-cell RNA on that. But in that time, 2016, you can also do DNA linked reads, and that was used for scaffolding. And then we had another set of genomic revolution when the long reads became extremely accurate. And now we have the PacBio hi-fi reads, which are their quality is higher than Illumina's. But imagine Illumina read that is spawning up to 20 kb. So these are the hi-fi reads that we are using. And now the recipe is not 30x per allele anymore, it's only 15x per allele. In some cases, you don't need that much. In some cases, when you work with polyploids, you might need more. But anyhow, 15x per allele. Oxford Nanopore current recommendation is 30x per allele, and you might need the Illumina scheme. Different organisms act a little bit differently, but currently in the BG, our solution is 30x per allele with current nanopore chemistry, and also add low coverage Illumina scheme. And then on top of that, you put something that is called chromatin conformation capture or Hi-C. And just to give you a little bit of the example of, still my mouse is not working, despite the retake. All right. Where to check all those beautiful recipes? You go to the QR code that you can see on the bottom of the screen. Now let's move to the right side of the screen, and you can see two different graphs, right? This is a human genome. On the left side, you see these beautiful yellow and blue bars. And that is something that has been created by a combination of PacBio, Oxford Nanopore, and Hi-C, that is so-called the telomere-to-telomere human. And then on the other side, you can see these stapled bars of black and white, where white represent gaps or missing sequence. And that is something that was used with traditional Illumina. That is the Human Genome Project data. So you can see that during this past, I don't know, that's like 10, 20 years, the quality of reference genomes have improved dramatically. So now finally we're making these beautiful chromosome scale references, not just for human, but actually for everything else, or majority of life. To say everything would be exaggeration. So just to iterate, the current recipe is 15x PacBio per allele, or 30x Oxford Nanopore per allele, plus low coverage Illumina only in case if you are doing Oxford Nanopore, right? On top of that, you put 50x coverage with Hi-C, and there you're free to use any method you want. ARIMA Hi-C, Dovetail Omni-C, if you are so inclined, Pore-C. They are comparable. There are small differences between them, but they work quite well across the whole span of the organisms. And after that, after putting the genome together, you optimally want to annotate that genome, something that Thomas will tell you a little bit later. So, and for that, there are different recipes, but the bare minimum accepted by the BGE and also EBP is to use 50 million reads per tissue mix, right? If you have a larger organism, when you can take out different tissues like vascular plants or animals, in some insects as well, then you might consider going as high as 100 million reads per tissue and then sequence as many tissues as possible, because that would give you a much better annotation or functional annotation at different tissue levels. But at the same time, it's more expensive. So think about what exactly are you going to use this genome for, if you have money to do that, if you're going to create reference for everybody and so on. And the good thing with RNA-seq, optimally, you would like to have it from the same organism, from the same specimen as you have the genomic data, but you do not have to. So somebody else from a specimen from another location could do the functional annotation using RNA-seq data and the reference that you have achieved. So perhaps not everybody in the audience are familiar with the Hi-C. So this is something that people ask very often about, say, what DNA quality do we need to have for Hi-C? And that's a misconception, because for Hi-C, we do not start with DNA. For Hi-C, we are starting with the tissue. And the quality of the tissue for Hi-C is even more important, the quality of the tissue for long-read sequencing and extraction of high molecular weight DNA. Why is that? Because we want to have the intact nuclei, right? Why is that? So you know that in all the genomes, we have something that is called chromatin structure and something that is called methylation. So you know the DNA is tightly wound against the histones, creating a nucleosomal complexes. So otherwise, yeah, if we take human genome and just stretch the DNA, we will go through the entire solar system. But somehow DNA that is as long as solar system can fit into the single cells in our body, right? So that is due to a very tight conformation of the nucleosomes in the cell nucleus. Also, why is it important to know how DNA is wound in these different nucleosomal complexes? Very often, when people look at the genome, for instance, they find a gene here, but regulatory elements for these genes, they can be found here. So why is that? Why so far away? It's because when they're sitting in the chromosome, they're actually in very close proximity. So it's sitting around the histone here, and then there is a loop, and that is sitting here. So we are trying to find out in this mess of different wound structures, within chromosomes and also between the chromosomes, how different portions of the DNA or portions of the genomes, they interact with one another. And that is really important if we want to do something that is called scaffolding, or to assign different reads to chromosomes. And how we do that? So we have the intact nuclei, really fresh, really well-preserved ones. And we cross-link our DNA to the nucleosomal complexes because we want to know which parts of DNA, which molecules are sitting together in very close proximity. And then what we do, we just chop everything and we leave only these nucleosomal complexes or the protein and then small DNA ends sticking out. After that, we do the end repair and trying to bridge them, right? So different DNA molecules sitting across the same protein, they will be merged together. And then we make a library from that and we sequence those chimeric constructs, and then we'll know that this and these portions of DNA, they were joined because they were sitting around the same nucleosome. And that is extremely important information that will help people like Thomas to put all these genomes together and to arrange that in different chromosomes. And he will talk much more about that later today. And finally, before we go to the next lecture, I want to address a couple of FAQs of frequently asked questions. Very often people are coming to us and say, yeah, we did an assembly of this organism 2015. It's really bad, and I want to improve it to a reference quality. It's not just my personal opinion. That is a common sense and something. It's when you're speaking to the sequencing professionals, bioinformaticians or genome sequencing professionals, you would get the same answer from anybody. So do not waste your time and just do it from scratch. Why? Theoretically, if you have a bad Illumina assembly, you want to improve it. If you need to use the same individual, right? How many of you have exactly the same individual in your freezers? And the quality of that is as good as it used to be 2015? You should not ever use another individual to create a reference genome if you have already data from individual from long before. So you want to sequence and assemble two alleles, not when putting one diploid organism and another diploid organism in the same soup. You have 4 different haplotypes. You will not make your life easier. You will just make it much more harder. So you need to use the same individual. But also, theoretically, if we'll add longer reads from the same individual, bioinformatically, it will be much more difficult to put everything together, error correction, and so on. And also the long read technologies, they are not the same as they used to be. So error profiles are completely different. If you had old CLR PacBio data with lower quality, and now you have beautiful hi-fi data, I mean, it's really not worth it, because for bioinformatician, the time that the bioinformatician will have to invest to work with this data set is much grander as compared to the price of the sequencing the way it is now. So you have to pay your bioinformatician the equivalent of 10 times sequencing the genome just to solve the problems. So it's much cheaper to sequence and make your bioinformatician very happy. And another thing people ask very, very often, oh, should I polish my hi-fi reads with Illumina? God, no. Absolutely not. Because you just introduce extra errors and it will not be good. So Thomas might cover that later in your lecture, but please do not polish hi-fi reads with Illumina data. Another question that comes up very often, how do I know if somebody else is sequencing my genome? Do I really need to create this reference, or somebody else is doing that? Or am I going to be scooped? So, and for that, we have an amazing tool available now, which is called GOAT, or the Genome on the Tree. There is a QR code to go there. So GOAT is a brainchild of Mark Blackster, who is a director of the Tree of Life project at the Sanger Institute. And for a long time, we called it Mark's Magic Database because people wanted to know how do we choose which genomes to sequence, especially in the Earth Biogenome project? How do we know that we're not duplicating the effort, that somebody else is doing exactly the same thing? So, and it started with pretty simple database of all genomes that are sequenced, and now with help of Sibeli, who is a project lead on the GOAT, and there is a link, GOAT has developed quite a bit, and it continues to be developed. You can search GOAT in many different ways. This is the front page, and there are tons of tutorials, and even more will come. The GOAT has tons of different functionalities, but today I will show you a very simple example. So there you see the arrow, and there you can type any taxonomic name of your favorite organism. So for instance, that is the species I worked on, that's distribution and ecology when I was doing my Bachelor of Science. And that is a CP, and I wanted to know if the genome of the species is existing. So look at the green again, and it says that the assembly span bar is red. That means that that genome is not complete. It will give you the information sort of about the genome size, but we also see that the sequencing data that we have, they are not in the chromosome scale. We also see the size of the genome, yes, it's 6. And we also see that the assembly, which is 5, is not covering the entire genome. We also know the chromosome number, and we also know the haploid number. Also, you can see that there is one species people are working on, and you also see that there is one project that is working on this particular species. If you want to find out which project it is, you click on that one project, and below you can see that this is Darwin Tree of Life. And you can also see that they have collected the sample. And yes, so and it's somewhere in their sequencing pipeline. For more information how to decode all these different flags in GOAT, there are tutorials online. Just go and look for it. The next example is something that a group at Uppsala University is working on. That's a herring, the common herring. And there, if you look closely, you can also see that it's in the chromosome scale. So the bar is not red anymore with some weird number. It's a chromosome. So that means that that's a genome of really good quality. Also, you can see that assembly covers a lot of things. And you can also see that there is one species. There are 35 different kinds of assemblies, but only one is chromosomal. And you can click on that chromosomal assembly and get much more information. And you can also see that there are 5 different projects who are working on that. You can also see to the right that there is a blob toolkit, there is a link to ENA, there is a link to NCBI, to the data sets, and so on. And further going down and clicking on different portions of the GOAT, you can also see which projects are working on it and so on. So why is it important to know which projects are sequencing that particular species? Because you can collaborate with them. It's not that they just assign these genomes to themselves and now they're going to sequence them by themselves. You can say, guys, hi, I have a RNA-seq set if you would like to use. Or, guys, can I collaborate with you? Or I have the tissue from here. Or I already have ongoing project that I did not declare on GOAT, but I have some data. So should we start working together on it? So why not? Another very important thing that I wanted to bring your attention to is the genome size. Genome size that you see on GOAT is not written in stone, right? So, and genome size attribution in GOAT, it can be erroneous in many different cases. So, for instance, in this particular case, which is the Clupea harengus, right? You can see that following the red arrow, you can see that these information of the genome size and c-value, it has the red underscore. That means it's inferred from an ancestor. It is information typical for a known sequenced ancestor of that particular group. So that is extremely rough guesstimate, right? And here is a classic example of noble crayfish, or Astacus astacus. In goat, ancestral reconstruction says that the genome size is around 4.5, 4.7. So when PI or researcher came to us and said, this genome is 4.7, please sequence it to me. And our target was to sequence 15x per allele, right? So that's, and we started sequencing at that number of smart cells that we have calculated we need. And then we're assembling it, and it's too fragmented. Okay, we think that perhaps the genome is larger. So we sequence double as much. We're looking at the K-mer plots, we're looking at the assembly quality, and still we're not reaching the saturation that we want to see. And then we reached out to PIs and said, guys, your genome seems to be much larger. We need to generate more data. And they said, yeah, okay, we have money, you can generate some more smart cells. And now our estimate of the genome size is at 12, right, of the coverage that we have sequenced. We're sequencing more smart cells, thinking that now the genome might be 12, and still we're not reaching saturation. So we're asking PI what to do, and they say, sorry guys, we do not have money to sequence more of the same individual. So this project is currently on ice. So that is a very typical example, especially in Malacostraca, especially in the crayfishes, in crustaceans, let's say, when the genome size is severely underestimated in the goat. So that is something to be extremely mindful of. So if we wrap everything in the sack and conclude this very first lecture of the day, let's say that currently, to address the biodiversity crisis, there are tons of different biodiversity genomics projects. And part of them is reference genome projects, because we want to create this huge global resource to enable any kind of biology to be studied at a completely different level of resolution. So in Europe, we have the ERGA, which is the European Reference Atlas and a regional branch of the Earth Biogenome Project. And we have also this BGE project, Biodiversity Genomics Europe. This is a part of ERGA, which teamed together with International Barcode of Life and trying to create this distributed network of biodiversity sequencing in Europe. Also, within these projects, we are trying to improve and to explain these different types of optimal workflows, and also for before and the downstream analysis. And that is the goal of this course. So we'll try to show you how to think and what to consider in all the possible different steps of the genome generation. And also take home from this particular lecture is that reference genome recipe, it is a moving target. So this is something that you need to find out or go and check quite often, because it's changing quite frequently. And also that everybody needs to be aware of goat. So you can find out if your species are sequenced or is going to be sequenced, so you don't have to do it, or you can see if you can find new collaborators, for instance, and do something together. But the problem with goat is currently that the genome size estimation is a little bit off target. And with that, I would like to say thank you, and we go to the next lecture.